screen here. So there are 2 summative assessments for the end of the unit. One of them which will drop at 12:50 today, and like I said, you can do it anytime before midnight. There's no time limit on the quiz, and people are going to be writing at home, so it's basically open book. Use whatever resources you'd like to complete it. It'll pop up in the little bar on the side, or you can click on assignments. It's called the Diversity Unit Summative Quiz. I will give you fair warning that I've never used the quiz system built into Brightspace before, so we'll call this a trial run. If there are some problems with the quiz, it doesn't work properly for some reason, I tested it. I did like a practice run through it and worked fine, but if something doesn't work, just send me an email, okay? And I'll fix it as soon as possible. It should be fine. I just never used it before. So it's 31 marks, I believe. It's a mix of true, false, multiple choice, and check all that apply. Just make sure that you are, actually looking at or answering the question like the correct way. So if it's check all that apply, don't just pick one. Or, sometimes it's only one, but sometimes it could be none, or it could be all of them, or whatever, okay? So just make sure you're answering the question type correctly. I've separated them out, so it should have a whole section that's multiple choice, and then a whole section that's check all that apply, and then a whole section that's true and false, so that it's not confusing. It's not like jumping back and forth between them or anything. Okay, so that's the quiz component at the end of the unit, and then the other part is the species spotlight assignment, which I'll go through the what it requires right now. How do I make it so that you? How do I do this? I need to show the student view. How do I do that? Can I do that on my iPad? No? Okay, let me switch over how I'm doing this here. You guys can see that? And I'm just going to make it so you guys at home can see that too. All right, how do I do that? Ooh. So I want display capture, but not this display. This display. There we go. Cool. Okay, so hopefully people at home can see that. All right, so if you look in the content area here, go to diversity, day 5, species spotlight assignment. There we go, perfect. So what you're gonna do, you're gonna pick a species. Any species you want, preferably not an animal. I said no animals, definitely no vertebrates, okay? Stay away from vertebrates. You guys know about vertebrates. You guys have seen vertebrates, you know what their structures look like, you know a ton about vertebrates. If you go outside vertebrates a little bit, normally I say don't do any type of animal, but I have to say that we abbreviated our little talk on animals quite a bit, so we didn't really cover the animal stuff as much as we normally would have. So you can pick an animal if you really want to, just don't pick a vertebrate, okay? No backbones. But other than that, any animal you want, okay? You want to pick a nematode, or you want to pick some type of mushroom or whatever, okay? Any organism. It could even be a single cell. It could be prokaryotic. Doesn't matter. Prokaryotes are tougher, by the way, because you may have you're going to do a dichotomous key, and doing a structural dichotomous key for a prokaryote is quite a bit more difficult. They look very similar. So you may have to talk about like what types of colonies they grow in, the shape of the bacterium. I think you're probably setting yourself up for like a much more difficult assignment to do. So maybe not prokaryotes, but it's up to you. You can do that if you want. It just makes it harder. Okay, so I previously did this as a poster, but I can't collect paper from you. So what we're going to do instead is we're going to do this as like a Google Slides presentation. So hopefully I modified everything here to fit into a slides presentation. You need a title slide. So tell me your name, obviously, and the course and the date. And the title is essentially just going to be your species name. Most of them have a common name, but not everything has a common name. So if it has a common name, then just tell me what it is. Like red squirrel, North American red squirrel or something like that. That'd be like its common name, although no vertebrates. And then give me its scientific name. So genus, species, right? It's a two-part binomial nomenclature. Use the proper formatting. It should be in italics. The genus is capitalized. The species moniker is not capitalized. Remember all this stuff. So species name. Okay, so that's what goes on your title slide. Then you're going to tell me a little bit about the taxonomic levels. Or the ranks rather, that apply specifically to your species. So let me pull up the little example at the bottom. So this is an example of what the poster looked like. You're essentially doing the poster, but each part of the poster you're just doing on another slide. Okay. So can I zoom in on this? It's gonna let me scroll. Okay, so this area right over here is explaining what is the taxonomic ranks for your species. So what domain does it fit into? What kingdom does it fit into? Which phylum does it fit into? And beside that, in a separate column, so you need another column over here, you're going to explain why it fits into that rank. Okay? For the first couple, it's very straightforward. For example, eukarya. Domain eukarya. Why is it in that? Well, is it prokaryotic or eukaryotic? Like, does it have cell organelles? Does it have a Golgi? Does it have a nucleus or whatever? So that's fairly obvious. Okay, so if you're picking a eukaryote, well, that goes in eukaryota. Then kingdom. Why is your organism specifically in that kingdom? So what makes it a protist? What makes it a fungus? Okay, we talked a little bit about that in class, about what defines something as each of those kingdoms. And so you're going to include that in this column. This is when it starts to get a little bit more tricky, okay? So then you're going to keep proceeding down through the ranks, and you're going to tell me why is it in that phylum? Why is it in that class? Why is it in that order? So in order to do that, you have to look up what that order is, okay? Usually there's like a Wikipedia article just on that order. And it'll say, this order contains organisms that blah, 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 have this, and this, and this, okay? Meanwhile, obviously the other orders there don't have that, okay? So that's the first part here. You're going to explain the taxonomy of your specific species that you picked, all right? And then in order to help you with that, I have some links that are quite useful. So there's two you can use here. This website right here is called the Animal Diversity Web. You can search for your species. It works better with species name than common name, although some common names will work here as well. And essentially this is a service to help you figure out the complete taxonomy of a species. So, use this. Not every single species is listed here, obviously. This isn't like a complete compendium of all of nature, but it's pretty good. So it contains a lot of stuff. If you're picking something that's very esoteric, like a species where there's very little information on it, and of course there's lots of species like that, recently discovered things that are of, minor scientific interest, you may be making your life very difficult. So what you might want to do is choose a species and then do a little bit of preliminary looking. And if you can't find anything about that species, in other words, we hardly know anything about it, maybe pick another one. Okay, that's not a great one for your project. So, but I would say for the most part, if you recognize it, like if you're like, oh, what's an example of a species that would just be, like if you're just going to pick like North American earthworm or something like that, something that's like really common, there's like a ton of information on that. You're going to find a million things. So probably if you recognize it, there's a million things on it. Okay, so that's that. There's also a second resource you could use here. Why isn't this working? There we go. Called the International Taxonomic Information System. And again, this is a compendium of living things assembled for the purpose of scientists to, catalog and figure out what the taxonomy is of a particular species. So you can search for stuff here as well. Not everything is in this as either, but it's pretty good. So you should be able to find some taxonomic information on your species here. So those are two good resources to use for that. If you still can't find anything on your species, switch species, okay? Pick something else. I would say if you can't find it in those two resources, it's a pretty esoteric pick probably that you've picked, so pick something else. All right, so that's the first component. Okay, a visual life cycle. You are going to show me a life cycle of your species. Normally I would get you to draw this. Okay, that's a little bit more difficult to do here. You could potentially draw it using some like Google drawing tools. You don't have to do that, okay? You can do it if you want to. But if you can find a general life cycle for your type of organism, it doesn't have to be specific to your species. But for example, if you're picking a flowering plant, find a flowering plant life cycle. One without an explanation of the life cycle, okay? Just the visual component. And then you are going to add in the textual explanation of the life cycle. Okay, so the important thing here is that you are going to explain the life cycle. Don't just copy and paste a ready-made flowering plant life cycle, okay, with everything typed in there. It's only the visual component that you can use. Normally I would get you to draw it, but obviously it's much more difficult now to do that. So you can use a picture of the life cycle for that type of organism, but I want you to explain it. Okay, so just add some text boxes in, explain the life cycle. If you need to use multiple slides for this because you can't fit all your information on a single slide, fine, no problem, okay? So the number of slides isn't really important here, it's just that you have all the information that you need. So I have an example of this too. Where is it? I opened that, didn't I? Oh yeah, right here. So there's an example of the life cycle. Normally this would fit on a poster, but you can make it a lot bigger, right? Because you got the whole screen here. These are on separate slides. So you're going to describe each part of the life cycle. Okay, next component. An image. Normally I would get you to draw this. This would be part of the biological drawing component of the course. We didn't do that. That's too hard to do under the current circumstances. So I'm going to get you to find a good picture of your species, okay? Which should be very easy to do. You can find a picture of it. But the second component of it is that you're going to tell me about some of the key anatomical features of your species, okay? So consider what the other species are in your genus, okay? And you can even go up a taxonomic level and look at the family level and say, well, what are the physiological features of the other families, okay, that are in your order? And so what is physiologically, morphologically special about your species? So for example, I picked humans here as my example. What is physiologically, morphologically special about humans relative to other species in our genus? By the way, all the other species in our genus have gone extinct. But of those, okay, humans had and have and continue to have a large brain to body size ratio. That's what distinguishes us from the other members of our genus. It's significantly larger than the other ones in our genus. As a primate, we have opposable thumbs. Okay, so obviously other families, not primate family, don't have opposable thumbs. We have external organs for sexual reproduction. Not all animals have that. Okay, so now I'm actually going up another rank here. But you're going to pick some key physical features that are obviously apparent from your organism that delineate it from other organisms. Okay, you could pick like body hair or whatever other things that primates have that other members of the family don't have. Okay, this is, by the way, a terrible picture. I'm sure you can find a real picture of your species. Okay, any questions about that so far? All right, next, you're going to create a dichotomous key. You guys have seen the dichotomous key. It's a decision tree. We made one way back 2 days ago or something in day 2. Or maybe it was day 1. You guys are going to create your own dichotomous key. This is obviously a blank one down here. But it's a decision tree that focuses on morphology. Okay? Pick 5 species total, including your species. So it's 4 species plus your species. Okay? Ideally, all of those species are in the same genus, but depending on what you've chosen, there may not be 4 other ones in your genus, so you may have to go up a rank, okay? So you may, for example, have to find 4 other ones in the same family. Rarely, there are not 4 other species in the same family. That's pretty rare. So if that's true, you can go up another rank and find 4 that are in the same order, but it's pretty rare. Usually you'll find 4 in the same family, okay? So the idea here is 4 closely related or relatively closely related species and your species. You're gonna put them, and then you're gonna create a dichotomous key to figure out, essentially if you had them all in front of you, how could you make a decision tree to decide which one was which, okay? Please use morphological features that are apparent from the picture, okay? If I can't see it in the picture, don't use it as a morphological feature, or just find a better picture of it that includes that morphology. Because the whole idea here is that when I look at the pictures, I can follow the key and get to your picture, okay? I test the key, that's the whole purpose of this. So if I look at the picture and I'm like, I can't see that, well then your key doesn't work. It's essentially, for my purposes, it's broken, because I don't have the organisms in front of me. So I gotta operate by what I can see in your pictures. So try and find a good picture that shows the features that you're talking about, okay? And obviously it's not gonna be a blank key. So you're gonna have decisions at each of these dichotomies, you know, yes, no, for each one so that I could follow the key. If you need to refer back to the note on this, we made a key for butterflies together. It looked just like that key, okay? Any questions about this? That's the last component. So what you wanna put on your very last slide is a works cited, okay? If you used an online resource to get this information, just cite it using APA citation, okay? The easiest way to do that is to use something like, you could use a Chrome extension. I use Cite This For Me. Cite This For Me is a great Chrome extension. All you have to do to use it is if you have an article like the nematode. Oh, I spelled nematode wrong. Okay, let's say you're using this article, okay? Wikipedia is probably not the best source to use, but you click Cite This For Me, okay? APA, and there it is. That's how easy it is, okay? It literally does all the work for you. You copy the citation and you put it on your last slide, all right? That's it. Some people are telling me that they can't get this to work. You can use citationmachine.net, or you can look on the library homepage and it explains how to make an APA citation. There's like a formatting thing you follow. You can see it down here. So there's like name of article, date that you accessed it in brackets, date that you retrieved it, web address, okay? All that stuff has to be included and explains the formatting. But I recommend using an app like this because then you can just skip all that. I'll be totally honest with you. So I went through all the way through university. I wrote a ton of research papers. I've never actually looked up how to do APA citation, so you don't really have to look it up, okay? There's a ton of tools that just automatically do APA citation for you, and you don't really have to, even in grad school I never used it. So just so you know, I mean, I cited all of my sources, you have to do that, but you don't really have to be super familiar with like what needs to be included. You just need to make sure that at the end your APA citation looks right. You know, is it clearly missing an author? Is it clearly missing the name of the article? Oh, something went wrong. I check it. But I mean, if it looks right, well, that's it. You got it. All right. So just as a heads up, there's a lot of tools you can use. You don't really need to know that stuff super well. Oh boy, some other science teacher's gonna really give me a hard time about saying that. But anyway, so that's the last page is your works cited. You always need to include that in any research project. Okay, guys. Images. Yeah, you're taking some images. You should probably cite those. You should include a citation for your images. That's not a huge deal. I really care more about the information, but technically you are supposed to cite images too, so you might want to include those. All right, it's a good habit to get into, certainly. Okay, before you get started, and I'm going to give you all of Learning Block 1 right to the end to work on this. You'll probably need every minute, and hopefully we can get through the genetic stuff fairly quickly in Learning Block 2, and I can give you the rest of the time to finish this. So, any questions about this? Yes?
Just to be clear, we're not presenting this in a video at all?
Nope. You're just going to submit this slideshow. So all you need to do is attach it to the assignment as a Google Doc and submit. That's it. Any other questions? We are going to do a type of presentation for the next unit, but it won't be to the class. It'll just be a short video that you're going to record and you're going to submit it, but not for this. We good? Okay, so the last thing I want to mention then is, I gotta switch this back. How do I do that? Can you guys see this? Yeah, great. So the last thing I want to mention is other stuff that's due today. You need to complete your diversity unit vocabulary list. So most of you have probably already done this. If you're done your vocabulary list, all you need to do is attach it to the assignment and turn it in. Okay, you may already be done, and it's already, I actually got it from several students already, but if you are done, turn it in. All right, those are the 3 summative things for this unit: the vocabulary list, the quiz, which opens at 12:50 today, and this assignment that you're working on. That's all you need to do for unit 1. I mean, it's not all, it's a lot, but that's what you have to do for unit 1. Okay? Once we come back from the break, if you're, just so you know where we're headed here, you can see the next notes are posted right there. I broke the next set of notes up into 2. So I have this package right here, which is, we'll call it review. Not all of it's review, but a lot of it is grade 10 review, which we're going to try and shoot through pretty quick. Most of this genetic stuff you would have covered. You guys talked about protein synthesis last year in grade 10? A little bit. The idea that DNA makes proteins, transcription, translation, just on a very general level. Okay, so we're going to do a review of that, and we're going to add a little bit more detail. And then we're going to hit the day 5 stuff here. This is what we're attempting to get done today. If by any chance you finish this project early, which you may not, and that's totally fine, feel free to take a look at these day 5 videos. You got to watch them at some point anyway. Okay, I'll give you the rest of the time. Good luck, guys. Let me know if you have any questions. Hey, guys. Welcome back. If you haven't done so already, for the second part here, you're going to need to open up the new set of course notes. If you look on the course homepage, now you got to go down to the second unit. So Unit 2, Genetic Processes. The first thing that's posted in there says U2, that's Unit 2, Part 1, Review and Genetic Processes Course Notes, Q1 2020. So you're going to want to download that, put it in your drive, and then open it with KME. So that's the new set of course notes that we're going to use for the beginning of the genetics unit. The genetics unit is broken up to 3 sort of mini booklets. The first one's all review, and then the second 2 are different aspects of genetics. So we're gonna break things up just a little bit. Genetics is probably the most complex unit that we're going to do together. Oh, also, for those at home, just if you wouldn't mind, it's just saying here or present, so I can check attendance again. Thank you.
Yeah, it's just everything's blanked out. All these, it won't let me click it. But it lets me get to the folder document.
Huh. That's weird. Can you show me your the course homepage, the very front page? I just want to make sure you actually are logged in so that you can add Google Docs. You should be able to do this. Like go to course home. Scroll down. Oh, no, you haven't done it yet. Link to your Google Apps account. Once you do that, and then there's another button under there after you do that that says authorize. You're going to click that again. Then you'll be able to attach Google Docs. So instead of clicking the attach file button, there should be another button there that's like attach a Google Doc, I think. Or you click attach file and then select Google Drive or something along those lines. I'm just going to take attendance real quick, guys, and then we'll get started. Everybody okay finding this note? Yeah? What's that? Yes. Oh, you do? Oh, okay. Okay. Oops. Okay, so what we're going to do now is a little bit of review from grade 10. Hopefully it's a review. Intermixed with probably what is a little bit of new information for you. This isn't technically part of the grade 11 biology genetics unit, but all of this stuff you need to know in order to do the grade 11 biology genetics unit. So most of it is supposed to be in the grade 10 curriculum, but it's okay. Like some of the stuff you don't get to, and sometimes you do little bits and pieces, but not the whole thing. So first of all, basic cell theory. You guys definitely covered this in grade 10. The idea that everything's made out of cells, right? All living things are made out of cells. We talked about this in the previous unit as well. You can get uni or multicellular organisms. The cell is the basic structural and functional unit of life. You can't get any smaller than a cell. And all cells come from pre-existing cells. That's basic cell theory. Some of the extra stuff here that we've added since the original cell theory is this idea of energy flow occurring within cells. So that's the idea that cells use some type of metabolic process to take in and release energy to use it within the cell. We're going to talk about some of the molecules that are involved in that metabolic process tomorrow, question mark, probably tomorrow. And then more importantly for our genetics unit, cells pass on hereditary information from one generation to the next generation. Sometimes that genetic information, that hereditary information, is modified as it's passed on to the next generation through mutation or through a mixing up of genes, which we're going to discuss how that works. But the idea here is that your DNA is passed on to your children, and the same is true of all organisms, including bacteria. They pass their DNA on to their daughter cells. And this probably came up during our diversity unit as well, but all cells have the same basic molecular composition. Cell membranes are basically the same in every living thing. The DNA and the way DNA is constructed is conserved among all living things, and that's because they're related to each other. But there's a conservation of these basic molecules that are used to compose all of that stuff between all living things. And tomorrow we'll get into what those molecules are, like the basic molecules of life. Okay, so last year you would have spent some time doing, it would have been a review of grade 8, I would think, of the organelles of the cell, right? Golgi, mitochondria, endoplasmic reticulum. This sounds vaguely familiar. Okay, so I'm not going to go through this with you, but once we're done the other part of this note, I'm going to get you to go back and read this. Okay, this is a basic summary of the organelles and what they do. Okay, it's a little coloring activity, so you can, if you want, go through and color these. I took some of the coloring activity from this biochemistry coloring book that I got in university. And the whole purpose of the coloring books, a lot of courses have these, especially in biology, is that it's supposed to like help you remember it. So like when you color it in, it like helps you remember what everything is. It didn't work that well for me, but for some of my friends thought it was really good. So I did have an anatomy coloring book that I used for all the muscles. So it was like, oh, color in the biceps. In one color in this. And that one actually I thought did help me. So I guess it depends. So it worked for me for some things and other things it didn't. But the coloring book concept does definitely help people remember things, certain people anyway. So you can do the coloring if you want. You don't have to color it. I don't really care if you do or not, but it may be helpful for you to help remember what these structures look like. So the idea here is that you're going to go through and name these. You should be able to name them based on the descriptions here that are given. And if not, lots of resources available to you. You can look online and stuff like that. There's like a million labeled cell diagrams that you can find. There's no shortage of this information. But this should all be reviewed from grade 10, right? There isn't anyone that's like, cell parts? Organelles, I've never heard of that. Okay, so this is a review. Great. So the reason that I need you to know this is because tomorrow we're going to talk about how those cell parts are involved in using your DNA to make proteins. That's called protein synthesis. It's one of the most important processes in cellular biology. And I'm going to be talking about different organelles through the whole thing. This organelle does this. Now this organelle does this. So if you have no idea what the organelles are, what they do, that's going to be a real tough concept for you, okay? So definitely do this review. There's also a video. What did I call it? It's posted up here as well. So it's on the day 5 stuff. Introduction to Cells, the Grand Cell Tour, okay? That's a video review of the organelles. You don't have to watch that one if you don't want to, but I would say that if you're like a little hazy on what all the organelles do, it's a pretty good video review, and it's not long. How long is this thing? 9 minutes, okay? So it's not gonna like hugely cut into your life, but it does do a pretty good job of reviewing what all the organelles do. And again, if video stuff is really helpful for you, that's probably gonna be a great tool. And then once you've done that, just go through and see if you can answer these. This should all be stuff from last year. I don't know. I don't know if you actually covered that. Did you talk about, for example, like specialized cells in the stomach, digestive system, islet cells and stuff? No? Maybe you did, maybe you didn't. Is it mentioned in here? I'm hoping all this is encapsulated in this reading. Oh yeah. Okay, so it's all of these are taken from the reading. So if you've done the reading, you should be able to go through here and answer these fairly easily. If you're really stuck, drop me a line, okay? I'll be available just like normal through the entire block 3, okay? People are regularly coming to visit, which is great. But I'm gonna move on. As a heads up, chapter 6.1, that doesn't sound right. Is that in this textbook? Oh, a little bit. So some of this information is in chapter 6.1 in your text. Keep in mind that a lot of this is kind of a mix of what you should have gotten from grade 10 and from grade 11. So not all of this is actually covered in the grade 11 textbook because it's meant to be a review. But some of this stuff is in chapter 6.1 your text if you're looking for more information. So we already mentioned this here. DNA is your hereditary material. We figured that out about 100 years ago. Didn't really know how to figure out what the code was 100 years ago. We figured that about 30 years ago. But 100 years ago, we figured out, oh, DNA is the thing. That is the thing that you pass on to your offspring. Before that, we thought maybe it's protein, maybe it's something else that we don't know what it is in the cell, but eventually we figured out that it's DNA. It's found in the nucleus of almost every cell, mostly. So there are some cells that don't have DNA in them, like your red blood cells. They don't live very long because they don't have the ability to produce their own proteins. So they basically have a set of proteins that they start with, and once those degrade, then the cell just dies. So that also exists, but for the most part, your cells have nuclei. Most of them only have one, although some types of cells have more than one nucleus, like your muscle cells. They're multinucleated. In terms of pieces of DNA, you have 46 pieces. They are independent from one another. So they're long segments of DNA, and those are called chromosomes. Did you guys talk about that last year? No. Okay, so that might be new information. Maybe, depending on the teacher. Okay, fair enough. So this is where there's like a bit of a mix. So you got 46 of those chromosomes. Hopefully, maybe you have 47. It's possible. You might not even know if you've got an extra one. We'll talk about that later. So, but chances are you probably have 46. If you take that complex DNA molecule and look at it very closely, by the way, you can't see this with a light microscope. It's barely visible with an electron microscope. So, but because it really is just like a single molecule, a really long polymerized molecule, but it's quite small. But it's a double helix. So a double helix is like a ladder. I'll draw it off on the side here. It's like a ladder. And then you've twisted the ladder. Not doing a very good job of drawing this twist here. And then you twist the twist so that you end up with something that looks like this right here. That's a double helix. It's a twisty ladder. I'll talk about what it's made out of in a second. And the structure of it is a repeating sequence of sugars and phosphates that makes up the sides of the ladder. Sugar, phosphate, sugar, phosphate, sugar, phosphate. Tomorrow we're going to talk about what a sugar looks like, but did you guys talk about that at all last year? It's like a circular molecule of carbons. That's a sugar. Yeah. And phosphate you would have talked about in grade 10 chemistry, right? Anybody remember what the formula is for phosphate? It's got phosphorus in it. And it's got oxygen in it. Do you remember? It's got a charge. Wait, is it 3 minus? Uh-oh. Oh man, everybody forgot their grade 10 chemistry, including me. That's terrible. What's the charge on phosphate? 3 minus. Yeah, I got it right. All right. So it's PO4 3 minus. Is that ringing bells for anybody? Hopefully it does from chemistry. That's a polyatomic ion, right? Yeah, from chemistry? All right, so PO4, phosphate. And then you got your sugar molecule, which is a string of carbons into a configuration that looks kind of like that. So you end up with this repeating pattern that looks like this. If you look on the right-hand side here, it goes sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. That's the backbone. It's just a repeating sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And then you have these other structures in the center that can differ. So these structures in the middle right here are called nitrogenous bases. So the middle structures are nitrogenous bases. And then you got on the sides here your sugars. The sugar, by the way, is a specific kind of sugar called deoxyribose. It's not glucose or anything like that, but it is similar to glucose. It's a modified glucose. Your body can make deoxyribose out of glucose. And then phosphate is just phosphate. So one group of those, like this, one of these groups right here, that is a phosphate, a sugar, and a nitrogenous base, is called a nucleotide. Okay? So if you take a bunch of nucleotides and you put them together, then you get a DNA sequence. So DNA sequence is just a bunch of nucleotides. And then on the other side of the DNA is just a complementary nucleotide. So there are four nitrogenous bases. Remember, those are the things in the middle. So the four are, I'll go back down to that in 1 second, adenine, guanine, cytosine, and thymine. And I mentioned that one side of the ladder is paired with the other side of the ladder. Well, adenines are always paired with thymines. So there's a thymine on the other side if there's an adenine on one side. And guanines are always paired with cytosines. So those are called complementary nitrogenous bases. They always stick together. They complement each other. So if one side is an adenine, the other side is a thymine. If one side is a thymine, the other side is an adenine. If one side's a guanine, then the other side is a cytosine. They can only chemically bond with their complementary pair, and if it's the wrong one, it physically can't fit together. The shapes don't fit. Yes? On these four names, I don't think I can zoom in anymore without having them go off the screen. Is that better? Yeah? Adenine, guanine, cytosine, and thymine. As a heads up to you, you need to know which ones pair with which. A always pairs with T, and C always pairs with G. File that away. You guys okay? All right. So if you look at this, you'll notice that you could just go along here and just read the code, right? Like you could just go, okay, A, G, C, T. Keeps going, right? It's a polymer. Keeps going and going and going. G, G, C, T, T, A, G, G, C, T, 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 A. It's a big, long code, okay? That is where all of the information is stored. It's all about the patterns of these nitrogenous bases. And tomorrow we're going to get into detail about how you actually use the pattern to make something out of it. It's a really cool process. But that essentially is where all the instructions are for how to operate your cell. It's just a sequence of letters. Okay? And then your cell can read those letters and do stuff with that information that's stored like that. One side of your DNA is the coding strand. That is, it has the code on it. And the other side is just a complementary strand. So the other side here is basically just there to protect this side. If you left these exposed in the center and they weren't bound to anything, you would basically have these like open chemical bonding sites where, you know, other molecules could just come by and stick to them, or some kind of rogue chemical reaction could happen and bust one of them off or something like that. So the other side here is basically like a protective side to protect the code. The other cool thing about having a complementary side is if you do, for some reason, bust one of these off, one of these nucleotides, like for example, let's say you go out in the sun and some UV comes in and blasts one of these off, which does happen, by the way, UV does that. Well, you have a whole bunch of enzymes in your nucleus that can go by and read and say, oh, we got a T over here, but the other side's missing. It knows for sure that if there's a T over here and the other side's missing, that it's got to put an adenine nucleotide in there, and it'll just come and repair it. So there's all kinds of repair mechanisms that go through and just fix those little errors, but it only works if you have that complementary side. So it's kind of valuable to have that. It makes it much more chemically stable. So we've got a coding side and a complementary side. If you're interested You can go to the Human Genome Project. It's on the Human Genome Project website, but it's also on it might be on the Human Genome Project website. Let's check. I can post a link to this if you want. Is it this? I don't think so. There's a website. I gotta figure out where it is. Where you can just find the whole thing. It's in a text file. So someone went through and said, okay, chromosome 1, here's the code from beginning to end. A, T, C, G, G, T, A, C, G. Chromosome 2, here's the whole thing from beginning to end. And obviously that's a useful resource because we can figure out which sections do which things, and they're called genes, by the way, so going back to our note here. A specific section of this genome, one that produces, oh, what happened here? One that produces a specific protein is called a gene. So if you go through that code and find a section that does one protein, this section makes testosterone. This section makes insulin. That section is called a gene. G-E-N-E. So when we're talking about your genes, we're talking about the sections of DNA that make proteins. And we figured out a whole bunch of these. Human Genome Project was super valuable in that way. We only finished the Human Genome Project, what, 7, 8 years ago? Not that long ago. That's when we first figured out what the entire human genome was from the beginning to the end. Okay, somewhere around when I started teaching, which is kind of cool. Not that long ago. And since then, we've figured out what a whole bunch of the sections do. This section makes this. This section makes this. This section makes this. Keep in mind that the genome that they used is one person's genome. So obviously, your genome is not going to be identical to the one that's posted on the Human Genome Project, but it is close. There's very little diversity actually within the genome. So, you know, your section for making insulin is going to be virtually identical to this person's section for making insulin. Minor modifications. Otherwise, you wouldn't be human. So, like, most of it's the same. Okay, so that's important. One section of DNA that codes for a single protein is called a gene. If you actually went through and read how many bases there were, there's about 3 billion. It's a pretty long text file, okay? It's 3 billion letters long. So that obviously, it took a while. I think the Human Genome Project took about 10 years to actually go through and figure out the entire code from beginning to end. Since then, we've genotyped many people. We can do it much, much faster. We can shotgun genotype a person. It's like a special technique for genotyping. You do it very quickly, and you can do it in like a week. You could figure out somebody's entire genome in a week. So it doesn't take that long anymore, but originally, our tools were pretty, they were new, and we didn't know how to, very effectively use them. So it took a long time to genotype things. So some interesting facts. If you just look at the genes, okay, the genes, remember, are sections that make proteins. You have about 30,000 sections that make proteins. So that's 30,000 genes, approximately. Most of the time, we can tell where a gene starts and ends because it has this very characteristic code in it that says this is the start and this is the end. So based on that, we can figure out that there's about 30,000 human genes, give or take. That also means, because it's 3 billion base pairs, I mean, a gene is not one base pair long, like it could be 100 or 1,000 base pairs long. But even then, that leaves a lot of sections of DNA that aren't genes. So those are sections that don't actually make things. Those sections probably are used to regulate the other parts. In other words, say, okay, turn this on, turn this off. And And that's a very complex system, how we regulate gene expression. But those sections that don't code for anything are called introns. So sometimes it's called non-coding DNA. That's the same thing. I think your textbook, no, the textbook's not that old. It may refer to the non-coding part as junk DNA. That's what it used to be called. But that was before we knew that it did anything. So it definitely does a lot of stuff. So this part of your DNA are called non-coding or introns. I think we mentioned the other day that bacteria do not have introns. So sections of their DNA that doesn't do anything, bacteria don't have that, but archaea do. So what that means is that only 1.5% of your genes, of your DNA, only 1.5% of your genome actually codes for something, like actually makes proteins. So a whole bunch of it does something else. That's the intron part. And I think this is really cool that we know the function for about half of them. That's really cool. So almost half of that 30,000, we know what that gene does, what it builds, what its importance is in the body. That's pretty good, I think, considering we've only been really researching it for maybe 10 years. I feel like that's pretty good. And we figure out what genes do all the time, new information about what genes do. So, how are they organized? Well, they're organized by size. So remember, a chromosome is just a section, a strip of DNA. And the first chromosome, we call it chromosome 1 because it's the biggest one, has the most genes in it. It has 1,961 genes. The smallest chromosome is chromosome 21, and it has the fewest genes, 2 to 300. Now keep in mind that there are 23 chromosomes total, because you have 2 copies of each. Not identical copies, but 2 copies of each. So that also means that chromosome 22 is smaller than chromosome 21 but has more genes on it. So that just means chromosome 21 has more introns, more non-coding DNA. I'll show you what they look like in a second, but they are organized physically by size. Any questions about any of this stuff so far? So every one of your cells has the same code in it. 46 strips of DNA, 3 billion base pairs total. And they have sections within them called genes that make proteins. And a bunch of other section that is intron that just turns proteins on and off. Make this protein, don't make this protein. Yes, we're okay with that? All right, basic genetic stuff. This right here, this picture right here, let's see if I can get the whole thing on the screen, is a human karyotype, okay? So this is an actual microscope image. You can take a cell from an adult that contains a nucleus. You can bust up the cell using some solution. You have to freeze it during mitosis. Do you guys remember mitosis? Yeah. So do you guys remember what happens during prophase? No, nobody remembers this. We're going to review this, by the way. So in prophase, you take that drawn out DNA that's laying all over your cell, nucleus, the chromatin, and you tie it up into these big sticks of DNA called chromatids. Does that sound familiar? You make your chromatids? Okay. If you freeze the cell right after it does that, basically the beginning of metaphase, okay, you can bust the cell, lyse the cell with some solution, and you can actually sort these. So you can take a picture of the DNA. They use a dye, by the way, to create this banding pattern The dye sticks to certain pieces, parts of the DNA pieces. You take a photograph under the microscope. These are with really powerful light microscopes. And then you can basically do a cut and paste. They do it with computers now, but a computer sorts all of those pictures of the chromosomes and puts them together into their chromosome number. Okay, so this is an actual set of somebody's chromosomes. You'll notice that there are 46. Assuming everything's going fine in there, that's the number you're going to get, okay? So I mentioned previously that, first of all, they're organized by size. So if you look at this, you'll see that chromosome number 1 is the longest. It has the most DNA in it. And chromosome 21 and 22 are the smallest ones. I think 21 is actually the smallest, so 22 is a little bit bigger. And then the last one, chromosome 23, is the sex chromosomes. So those are the ones that determine your biological sex. If you have 2 X's, that's female, and if you have an X and a Y, that's male. We're going to get into that a little bit later. Okay? So we organize them here by size. It's a little bit difficult to only organize them by size because some of them are similar in size. And so if they're similar in size, the second thing we do is we organize them by banding pattern. So you can look at the bands on them. They always match up between the 2 chromosomes of the same type. Those are called homologs. So homologs are the same chromosome, chromosome 1 or chromosome 2 or chromosome 3, but they're not identical to each other. They just look identical under the microscope. And then lastly, you can look at their centromere location. So their centromere is this part here, the little whoop. And that's when you make an identical copy of these in mitosis, they're attached in that X configuration by their centromere. You can see them on here. There's a centromere here, centromere here. Can you guys see that? There's like a pinched in part. So when you get those like X's, when you make copies of these in mitosis, that's where they're attached to each other at the centromere location. Okay, so the reason why your 2 chromosome 1s are not identical to each other is because you got one of those chromosomes from your mom and one of those chromosomes from your dad. Your dad and your mom are not identical twins. So their chromosome 1s are not the same. They are different. They have the same information on them. So they will have the gene for eye color, the gene for making insulin, whatever's on chromosome 1, okay? They're going to be the same genes, but they won't be identical copies because they're slightly different because they're from slightly different people, okay? So when you get 2 things that are the same genes but not identical, that's called a homolog or a homologous pair. That is a really important piece of terminology that's going to come up a ton in this unit. So that's one definition you're going to want to really nail down. A homologous pair is 2 pieces of DNA that have all the same genes on them, but they're not identical to each other. So they do the same thing, but they're not identical. So you have 23 homologous pairs. Well, actually, the sex chromosomes are not always homologous. If you're a male, you have an X and a Y, and those aren't homologs because the X chromosome and the Y chromosome don't have the same information on them. So, but if you're female, your X chromosome is homologous. So where do we get this stuff? You can do this with an adult cell. So I could just take a cell from your skin or whatever, liver or pick any cell that has a nucleus. Or more commonly, this is done with cells in the amniotic fluid. So that's called an amniocentesis. When you take a little sample of amniotic fluid from a woman while she's pregnant, you freeze the cells in metaphase so that you have those nice big chunky DNA sticks, the chromatids, and you put a little dye on there and take a photograph. So the reason that they do that in utero is to check, are all the pieces here? Is there a piece of DNA, one of the chromosomes, that is, there's something wrong with it? It's maybe only half of it's there. Maybe you have a double up of a section of one of your chromosomes or something along those lines. Okay, so you can tell a lot of genetic anomalies just by taking a look at this. This procedure used to be very common. It's not as common anymore. So now they can find out a lot of this same information by actually just giving a blood test to the mother. So you can avoid taking an amnio. There's risks associated with poking a needle into the amniotic sac. It's fairly low risk, but obviously there is a risk associated with it. So it's mostly avoided unless you really feel like it's necessary. So they don't do this quite as much anymore. But there are the 46 right there. So the reason on the previous slide, the reason it says you have 23 is because you have 23 pairs. Okay, there's one of each of the 23 plus their pair. So going back to what we were talking about in the diversity unit, do you guys remember I talked about haploid and diploid? Does anyone remember that? So haploid is half of your genetic complement. That's when we were going through diversity and we were talking about reproduction. We're saying a person or an organism has to make a cell that has half of the total DNA. That's haploid. So if this person wanted to make a baby, they would have to create a cell with half of these chromosomes. It's a pick and choose, okay? This one from number 1, this one from number 2, this one from number 3. It's random, but that would be haploid. So your haploid number, your 1N number, is 23. Okay, that's your haploid number, half of your full genetic complement. Your 2N number, or your diploid number, is your full genetic complement that you have as an adult, which is 46. Okay, so your 2N number is 46 and your 1N number is 23. And it's different by species. So again, I'm just going to go over this terminology here. There are 23 pairs. They are in numbered sets from 1 to 23, and that's also known as your haploid number, your 1N number. You have 23 pairs. They're arranged from largest to smallest. 1 is the largest, 22 is the smallest, and then they just kick the sex chromosome to the end. The X chromosome is actually quite large, but the Y chromosome is just a little teeny tiny one. Obviously, the Y chromosome isn't that important because, ladies, you don't have one and it doesn't really make that much difference. So you don't have to have a Y chromosome. Can't be that important information on there. So in total, you have 46, as I mentioned, and that's called the diploid number, or the 2N number. That's specific to humans. If you're a chimpanzee, you have a diploid number of 48. But it's always an even number, because it has to be double of the haploid number. So it's always going to be an even number. You can't have a diploid number of 11, because then your haploid number wouldn't be a whole number. That doesn't work. So 22 of those chromosomes, that's 1 to 22, are called autosomes. An autosome, sorry about the vocabulary here, guys, there's going to be a lot. An autosome is a non-sex determining chromosome. So most of your genetic information has nothing to do with determining your sex, and those are called autosomes. But you do have 2 sex chromosomes, and those sex chromosomes are called allosomes. We just put them at the end of the karyogram, even though they're not necessarily the smallest. So if you look back up here at our karyogram, you'll notice that this person has 2 X chromosomes and no Y chromosome. So that makes this individual female. Okay, if you've got 2 X's and no Y, that's female. If you have an X and a Y, then you would be male, biologically anyway. So I found a male one on the internet too, and I took a picture of that one. So it would look like this if it was male. It's a male sample. One X and one Y. And the Y is just a little bitty thing. Questions about this before we move on? These are just like basic genetic structural things. It's a vocabulary that's going to be important, so you're going to want to add that stuff to your vocabulary list. Okay. Let's talk about the process. Oh, hold on. How much time do I have here? Should we do this? Hold on. Let me double check, see what we've got to finish here. Okay, I'm actually going to call this. Because I think if we keep going here, you guys are not done your projects, are you? No, I didn't think so. Okay. So I think what we're going to do then is I'm going to give everybody an opportunity to complete the review of the organelles, okay? Because I think you're going to want to do this before we start getting into the next part, because I'm going to be referring to them all the time, and you got to know what they do. So I'm going to let you do this part. Read this over. You can do the coloring activity if you want. Label the animal cell. And then just answer these questions from the reading. This will help jog your memory about what all the organelles do, okay? So that's the first part. Then on the website, on Brightspace, I gotta figure out why that crashes every time. On Brightspace, watch this video, okay? Especially if you're still unsure about what all the organelles do, watch this little organelle video. And then, definitely before you come to class tomorrow, make sure that you watch these two, okay? So this is going to give you a visual representation of what happens when you create proteins out of DNA. It's a complicated process. Maybe watch the DNA transcription one. That's how does DNA or what is DNA and how does it work? Maybe watch it twice, okay? It's a complex process. Then tomorrow in class, I'm going to go through the process again, but it's much better, I think, if you see a visual of it happening before I do it together with you, okay? It'll make it easier for you to understand. And then I think we'll pick up again with the note tomorrow. So what you can do right now, finish the organelles component, watch the videos. If you haven't submitted your vocabulary list from diversity yet, do that. If you add new words now to your other vocabulary list, genetic processes vocabulary. So I created a new assignment. There's going to be a vocabulary list for every unit. You probably learned 5 or 6 new words just now, I'm assuming. Diploid, haploid, homologous, gene, deoxyribose. There's lots of stuff here you can probably add, okay? So there's lots of stuff from this note. So I would consider adding some of that stuff to your vocabulary list. Does anybody have any questions about the unit quiz that you're going to write in block 3? No? Okay. Anyway, if you finish that stuff, feel free to work on your project. If you have any questions about your project, I'm here to help you. And as soon as 12:50 hits, you will have access to the quiz. You can write it any time between now and midnight. Just do it today at some point, okay? You don't have to do it right away, and it is open book, all right? So use your notes, whatever resources you'd like to have open to you to write it. Okay, good luck, guys. Let me know if you need anything. Hello. Oh, there we go. Oh, sorry for the people at home. Sorry. I accidentally had my microphone muted. I'm going to do this again. I'm just letting you know for the people at home that we have had a confirmed COVID case at school. So, I don't know, you guys may already be communicating about this. It's posted on the board's website. There's no indication that there's been any community spread. So, as far as we know, it's just isolated to a single individual. It's not this class because we would have received a phone call if that was true. So I'm just letting everybody know that there has been at least one confirmed case in the school. So it's whatever we're doing at school right now to prevent transmission is important. People need to keep that in mind. Also, if you have any questions, I'd be happy to answer them. I may have answered them already. Sorry, my microphone was off, but I'll answer them for people at home too if people have questions. Yes. So the school would only be closed if there were at least 2 cases of community transmission. That is, when there's cases confirmed to have happened at school. And right now we have 1 case that probably wasn't confirmed as a community transmission case, I would assume. So no. You will be at school tomorrow. At least the school will be open. Any other questions? It is important that you're following all the transmission protocols when you come, though. Yes. The short answer to that question is if you want to participate from home because you don't want to come, your parents need to call in and let the school know why you're staying home. But yes. I actually don't know the answer to that question, Emily. So when somebody is diagnosed, like due to privacy concerns, like they don't tell us who that person is, they do call you if you are one of the people in close contact with them at school. So I haven't received a call. So it's not somebody who's been in close contact with me. And no one in the class has either, so it wouldn't have been someone in our class. The only thing that they tell us is that there's been a confirmed case. Check on the board website. Oh, it actually says on the board website. So they say on the board website that it's 1 student. So there you go. So if there is a case of community transmission, then public health decides whether or not they want to close the school, but it has to be confirmed community transmission from the school. So, for example, there could be another case at the school tomorrow where that student got it from not school, somewhere else, home, in which case, if they can confirm that, then they don't count that as a school community transmission. So right now, there are conditions for closing the school and transitioning into online learning, like complete online learning, would be if they have multiple cases of school transmission. And what they're suggesting is that if there's no confirmed community transmission within the school, that our current health and safety protocols are effective and there isn't community transmission happening here. So That's the theory anyway. Did that answer your question, Angela? Sorry, hope it did. This was something that was bound to happen, so I mean, it was pretty much guaranteed to happen. Okay. All right, so I'm gonna take back off here again and go back just to chat. So if you do have questions though, you can drop them in the chat, or if you feel like it's a private question or whatever you feel, you can just email me if you need to. Okay, but I'm available to answer questions about it if you need it.